Cool, we might start then. So, hi everyone, and thanks for joining us today. So, we're very pleased to have Dr. David Benkeser. So, David is a former grad student of our department. He got his Bachelor of Science and Masters of Public Health in 2010 from the University of Georgia, and then joined our department as a PhD student and graduated in 2015. He then went to Berkeley as a postdoctoral research fellow and is now an assistant professor of biostatistics and bioinformatics at Emory University. So his talk today is going to be on the design and statistical analysis of COVID-19 vaccine trials. By the way, a quick reminder to the audience, please put your questions in the chat and ask to be unmuted so you can ask your question directly. And yeah, David, thanks for joining us today, although virtually, and we look forward to your talk.
Thanks. Sorry, I'm looking at your notes. Okay. All right. Yeah, it's really great to be here, even though here is, in my office in Atlanta. I was kind of looking through the participant list and I see some familiar names, so it's nice to see everybody, if only through this virtual session. I was promised that Scott Emerson would not be in attendance, and I see that he's here, and so that's disappointing. But just kidding, Scott. Nice to see your name. So yeah, I'm going to give a high-level talk of sort of some of the statistical issues that we've been grappling with throughout the process of developing the phase 3 trial protocols for COVID-19 vaccines. And of course, this talk represents, hardly any of my work. This is work in conjunction with a lot of statisticians who are out there in Seattle at the Hutch, Peter Gilbert, Holly Janes, Yunda Huang, Alex, Marco, the list goes on and on. So really, these are not my ideas, so I won't take credit for them. And if anything angers Scott, then I'll ask him to deflect blame onto Marco or another suitable candidate. So it's a high-level talk. I'm not going to get too much into math. I think I have 2 equations, and they don't come until like the second to last slide. So it's really sort of a thought piece to go over some of the issues that are challenging when you're thinking about how we should design these trials to evaluate COVID-19 vaccines. And so very happy to hear any feedback or thoughts you have on this. You can, contact me via email. It's just my last name, Benkeser @emory.edu. I got a Twitter recently, which has been a wild experience being on Twitter and seeing that. So you can follow me on Twitter. And then if you want to access these slides, they're available on my GitHub. You can just head to this URL that's down at the bottom and follow along. And I've hyperlinked some references there, so that's easy for you to pull those up along the way. Okay, so again, it's a high-level talk, and I'll start by giving you my biostatistical or biostatisticians' understanding of SARS-CoV-2 and actually the biological mechanisms of some of these vaccines. And so we'll start just by talking briefly about, how does SARS-CoV-2, the virus, go about infecting your cells? What's that process look like? Because that'll help us understand sort of where the vaccines are trying to target and prevent infection in clinical disease. And so SARS-CoV-2, like many viruses, is sort of depicted here. So I've learned that Washington Post is actually able to make prettier graphics than I am able to in PowerPoint. So I've stolen their graphics here to go through these slides. And so SARS-CoV-2 is here. It's illustrated on the left side. And like most viruses, it's a little bit of genetic material, RNA, that's encased in a glycoprotein. And so here the protein, that's these little knobs that are sitting on the surface of the virus. And one in particular importance for vaccine development is the spike protein. Okay, and so why is the spike protein so important? Well, it's important both for how the virus replicates in the host and as well as where we're trying to attack the virus with immune responses induced by vaccines. And so how this virus infects your cells is this little spike protein, and this is, again, a biostatistician's understanding of immunology, so probably saying many silly biological things, but my understanding is this little spike protein basically grabs onto your cells through this ACE2 pathway, the ACE2 receptor in your cells, and thereby admits itself into your cells where it releases its RNA, that gets translated into protein, so it's tricking your cells basically into replicating itself, and then those pathogens, those viruses are released back into your bloodstream to continue to go infect more cells. And so how do we go about trying to stop that with vaccines to develop immune responses that stop that process? And so we'll give you a little bit of background on sort of immunology, again, from a high level. And so I'll go through a few slides about how these SARS-CoV-2 vaccines are constructed. And in each of these diagrams, sort of the left side is going to be different and the right side is going to be the same. So we're going to start on the right side to talk about sort of what are we aiming for? What kind of immune responses are we hoping these vaccines can generate? And then we'll move our way over to the left side where we'll talk about sort of how do we get there? What are the different components of the vaccines that we can hope induce these immune responses? So starting on the right side here, there's really kind of two key immune responses that we can hope to generate. One is a T cell response, and that's really just generating cells, T cells, that can eliminate virus infected cells. So they're able to detect cells that are infected by these viruses and directly eliminate those cells. And the second one is perhaps more important or sort of a more focus for the time being in the vaccine world is antibody responses. So antibodies are made by B cells, and these are these sort of Y-shaped molecules here. And those antibodies, what they do is they basically grab on to the surface proteins of the virus. And so there's kind of two types of antibodies that we think about for vaccine development. There's so-called neutralizing antibodies. And what those are going to do is basically latch onto that spike protein. Remember, the spike protein is what the virus is using to grab onto your cells and insert itself in there. So what these antibodies are going to do is basically grab onto those spike proteins, thereby neutralizing the virus. So if the antibodies are sitting on those proteins can't be grabbing onto your cells. There's also binding antibodies, which as far as I understand, basically don't neutralize the pathogen per se, but they grab onto the pathogen and then they recruit other cells in your body to help neutralize the virus. And so those are really the immune responses that we're trying to generate by giving people a vaccine. And there's sort of this middleman here, these antigen presenting cells. And so the idea there is that these are the cells that see a little bit of this pathogen. They see the spike protein, right? And then they sort of train your immune system to recognize that. So these are the guys that are telling your T cells and your B cells, hey, this is what this virus looks like. So that those cells are able to produce a specific immune response that's able to target and attack specifically SARS-CoV-2. And so the whole game here with viruses is basically getting a little bit, some little bit of the SARS-CoV-2 virus to these APCs, these middlemen, right? And so the big difference in all of these vaccine constructs is how they go about doing that. And so the first one I have here is so-called nucleic acid vaccines. And I present these first because these are sort of on the front wave of the vaccines that may be approved for SARS-CoV-2. So the Moderna vaccine is an mRNA vaccine. Pfizer's vaccine is also an mRNA vaccine. And these vaccines are pretty neat. This is a pretty new technology. And the basic idea is that we're wrapping a little bit of mRNA into a lipid shell. And that's what the vaccine is constructed with. And when you're injected with the vaccine, that lipid shell is delivering mRNA to your cell. And it's basically tricking your cell not into producing a virus, but producing just the spike protein. And so basically your cells are kind of like the workhorse here. They're the ones that are generating the antigen, the spike protein, that's then released into your bloodstream, right? And that's what's presented to these APCs. Okay, and so this is a novel technology, never been used in an approved vaccine before, which isn't to say that it's been tried many times and failed. It's just to say that it's a brand new technology, and so it's sort of exciting in a way, and scary in a way that we're evaluating these vaccines for the first time in the context of COVID. So why do we like RNA nucleic acid vaccines? Well, there's quick manufacturing. We'll talk about some of the other vaccine constructs and learn that if you try to produce these spike proteins on your own, that's actually somewhat of a time-consuming and labor-intensive process. So if we're making those on our own and putting those proteins directly into the vaccine, that's pretty challenging. And so what's cool about these RNA vaccines is that they're very quick to manufacture, don't necessarily require a cold chain, so very important when thinking about delivery of these vaccines, particularly in the developing world. But again, this is brand new technology, never been approved before, and I think you can find some people in the vaccine world that are highly skeptical that these vaccines are even going to work. So it's sort of interesting that these are the first ones that are primed to deliver results from these phase 3 trials. So sort of more classical constructs of vaccines, so viral vector vaccines, the idea here is we're basically taking a little bit of the SARS-CoV-2 genome, some bit of that, and putting it inside of another pathogen, inside of another virus. And so when you're given the vaccine, right, that other virus infects your cells, and the genetic material we've kind of spliced into that other virus is read by your cells, and that's used to produce the antigen. And so that's the process that's going on here. And so these are sort of the second wave of phase 3 trials that are getting launched. The AstraZeneca vaccine is with this construct. So that's the Oxford vaccine, you may have heard it called. And Janssen, so Johnson & Johnson's vaccine. That's also a viral vector vaccine. And so the vector of choice for these is an adenovirus. In AstraZeneca's case, is a chimpanzee adenovirus. And this is a virus that just is associated with the common cold. Okay, and so again, we like these because it's a tried and true technology. We're able to quickly manufacture them, scale up manufacturing, and that's a good thing. One of the challenges with these vaccines is that it's actually possible for you to develop immunity against the vector, right? So you can develop immune responses against adenovirus, right, that are so good that it actually prevents the adenoviruses from infecting your cells and producing the antigen. And so oftentimes with these viral vector vaccines, we'll see that individuals will develop immunity against the vector, thereby leading to sort of waning immunity to SARS-CoV-2. Subunit vaccines. So this is what's going to be a Novavax vaccine and Sanofi Pasteur. And the idea here is that we're going to grow these protein subunits. So these spike proteins, for example, are going to be grown in a lab. So I think in Sanofi's case, they're produced in insect cells actually in a lab. So they're infecting, as far as I understand, infecting insect cells with SARS-CoV-2, getting them to produce this spike protein, and then isolating that spike protein and building the vaccine in that way. So in this sense, we're just giving you directly these proteins as part of the vaccine. And this is a mechanism that we've created several effective vaccines before. The hepatitis B vaccine is constructed in this way, HPV, and several other highly effective vaccines are subunit vaccines. So it's kind of a well-studied way of constructing vaccines in terms of safety profile and efficacy for several viral diseases. The downside, of course, is it's a bit slower to manufacture, right? This process of growing these proteins in the lab, of purifying them, takes some time. So it's a bit less scalable, perhaps, than some of these other technologies. Another challenge is that often these vaccines don't always induce a very strong immune response on their own. And so you often require an adjuvant. And so an adjuvant is just sort of a little something extra, basically, that you put in the vaccine beyond just these protein subunits. That's really there to try to give you more of an inflammatory response, to kind of wake your immune system up, say, hey, you need to send some cells over here, there's something going on. Okay, finally, there's sort of, I think what probably my grandparents think of all vaccines is the weakened and activated vaccines, right? So that's where we're intentionally weakening or injecting you with an inactivated version of the virus. And so that's the construct of several, classic vaccines like polio, flu, MMR. Some highly effective vaccines have been designed in this way. Again, slower manufacturing, right, because we have to go through the process of cultivating these viral strains in the lab and weakening and inactivating them. Again, also requiring an adjuvant. And so none of the US phase 3s, as far as I know, are using this construct, but some of the Chinese manufacturers like Sinopharm and Sinovac are. Okay, so there's your crash course in immunology. You now know exactly as much as I know about SARS-CoV-2 immunology. And so we're primed to talk about more statistical issues, the vaccine development process. And so this slide is taken from a nice paper in New England Journal, sort of highlighting what's changed from the traditional paradigm of vaccine development and approval to what we're trying to achieve, in the context of a pandemic. And so on the top portion of this figure is sort of showing the traditional paradigm for development. And not only, the sort of clinical trial process that it has to go through, but also the way that the manufacturers are thinking about scaling up manufacturing. Because they're both of course, important components of delivering an effective SARS-CoV-2 vaccine. We have to prove not only that it's safe and effective, but also be able to scale up the manufacturing such that once it's approved, we'll be able to deliver it pretty much to everybody around the world as fast as possible. And so I think, we're all biostatisticians, so many people are familiar with the clinical trials development process. So I'll just say that briefly, maybe for the younger grad students who maybe haven't been exposed to this. So the idea in the traditional process is a very deliberate process of clinical evaluation, right? So there's a lot of sort of preclinical work that goes in to figuring out how to construct these vaccines. And then we enter what's called phase 1 trials. And phase 1 trials are typically, finding appropriate dose, establishing some safety, first trials in humans, right? So you're giving the vaccine to a small number of people and basically showing that it does something and it doesn't kill everybody, right? So that's sort of the phase 1. And then we roll that into a phase 2 where we're giving it to some more people. right? And maybe phase twos roll into like phase 2Bs, which is more like a randomized study of efficacy, but usually in a smaller population, maybe a couple thousand individuals, targeting very high risk individuals. So you want to start seeing some efficacy signal in a phase 2 trial, right? But they're deliberately sort of underpowered, or they're in a more restricted population than you're hoping to get the vaccine approved in. And if you see, good signal from phase 2, that's when we hit the big boy, right? We run into the phase 3 trial. That's a huge trial, a licensure trial, right? Where the results are going to be submitted to FDA or whatever regulatory body there is, and they're going to carefully scrutinize the efficacy signals, the safety signals, and if everything looks good, we'll grant licensure for that vaccine to be released to the public. And so just to note, right, that on the top here is the manufacturing process. And so in traditional paradigm, right, companies are very hesitant to start large scale manufacturing before they know their product's going to get approved, right, which makes a ton of sense. They don't want to invest a ton in scaling up their manufacturing setup until they're sure that they have a marketable product, right? So really that large scale manufacturing isn't happening in the traditional paradigm until after licensure, right? So what's happened, in the context of a pandemic, in the context of the current pandemic, in terms of how we're thinking about trying to compress this timeline to get a vaccine delivered more quickly, well, all of this has basically been compressed, right? So phase 1 and phase 2 trials have sort of been combined into single trials. The phase 3 trial actually hasn't changed too much, right? And that's something that I like to emphasize, right? So there's sort of this question that's asked all the time of reporters and people, the general public, is, are we moving too quickly, right? Are we missing out on crucial safety and efficacy data? And I would argue that no, the phase 3 trials that we're doing look very similar to phase 3 trials we would do even outside of the context of a pandemic. So while the preclinical development phase has been compressed, while the phase 1 and phase 2 has been compressed, I would say that the phase 3 is still proceeding sort of as we would expect and up to the rigorous standards that we would expect of a licensure trial. Now that said, the manufacturing is really the paradigm has really shifted in the context of a pandemic, right? So one of the things that we want to be prepared for is large scale manufacturing pretty much the moment, right, that the licensure is granted, and even before that. So that's what's been happening, is these companies are not only putting these vaccines into phase 3 trials and manufacturing enough of the product for the trial, right? They're already thinking about large scale manufacturing of this, okay? And so this is just a chart that's showing, the vaccine development, typically takes a very long time, right? So some of the classic vaccines, are on the scale of several years, tens of years, 30 years for chickenpox, right? Malaria is probably about 30 years also before the first vaccine was approved. So this is a process that usually takes a very long time. And so for COVID, we're really hoping to achieve this in under just a couple of years, which would be a pretty extraordinary achievement if we're able to do it. And so how has the US government been involved in this? Well, there's sort of two ways, and this is like a terrible Edward Tufte non-approved graphic, so please don't spend too much time looking at it. There's a couple purposes here. One, to get me to talk about Operation Warp Speed. What is it? Why does it have such a cool name? And what are they trying to accomplish? So Operation Warp Speed, or I'll call it OWS, right, is our federal government's response to trying to help speed up the vaccine development and approval process. And so there's really two ways that I think that OWS has tried to go about this. The first is by engaging companies to basically say that we'll pay for you to run your phase 3 clinical trial. So we'll assume the cost of that. So they enter into an agreement with BARDA, which, again, this is my biostatisticians' understanding of how all of this works. They enter into agreement with basically a branch of the NIH. And BARDA says, we'll give you money to fund this phase 3 trial. And the government will have some oversight in terms of how that trial is conducted. And so that's sort of how we end up talking to these manufacturers and sitting on some of these calls to advise them on how these trials should be designed, is through these arrangements with BARDA. And that's what kind of gets our group of statisticians, our government statisticians, as representatives on the design of these phase 3 trials. So that's the first way, right? We can either, we can help them directly run these phase 3 trials, we can pay for them. The second way is that the government is paying for manufacturing, right? So the government is basically assuming the risk here by basically pre-ordering a large number of doses of these vaccines, irrespective of whether the vaccine is eventually found to be effective. Okay, and so that removes the risk from the company, motivates the company more to put the trial into field, to ramp up the manufacturing even before they have an efficacy signal. And so every sort of company that's seeking approval for a vaccine in the US has taken advantage of that manufacturing route. So they all have manufacturing agreements with the US government. And then most everybody except Pfizer has also entered into an agreement with OWS to help design their phase 3 trial. Okay, so Pfizer is sort of the oddball in this bag. So there's a manufacturing agreement with Pfizer, but not this sort of oversight coming for their phase 3 trial. Okay, so where do all our colleagues at the Hutch and UW and some of who are sitting on this call, where do we come in? It's through this COVID-19 prevention network. So CoVPN, I'll call it. It's a network, clinical trials network formed by NIAID to establish a unified framework for getting these trials into the field, for evaluating these vaccines and hopefully eventually monoclonal antibody studies as well for prevention. And so the network was formed by pooling resources across existing trials networks. One of them was the HIV vaccine trials network, right, which is where a strong presence from the Fred Hutch exists. And so this group is really a bunch of clinical sites, laboratories, people who specialize in recruitment and retention for clinical trials, and of course, we statisticians. And so what sort of role have we been playing as statisticians on these trials? It's been sort of an interesting process, I would say, of sort of this agreement that these trials or that these companies have with the government, right? It's been sort of interesting in terms of who they listen to, because they're getting feedback from a lot of people. They're getting feedback from FDA, they're getting feedback from us, they're getting feedback from statisticians at NIH. So there's this huge sort of collaborative effort, and nobody's really sure who has the authority to tell these companies what they can or can't do. So it's been sort of a learning process for all of us, and an interesting last few months working with these companies. So what have we been mostly working on? So we sort of provide advice, guidance, suggestions on how to do the primary trial design and analysis. And that's somewhere where we've sort of made suggestions and sometimes firm suggestions, and sometimes the companies listen to us and sometimes they don't. So there's definitely sort of the companies still have some leeway in terms of designing the trial and the analysis plan that they want to try to bring their product to market. And we're there sort of as an advisory role for that. One place that we've sort of had a big say on some of these companies that maybe aren't used to bringing vaccine products to market, like AstraZeneca hasn't run a vaccine trial in over a decade. So they're sort of interested in hearing on how that's currently done. So we've been providing input on sort of how to do sequential efficacy monitoring, how to do safety monitoring. We help them address comments that come back from the DSMB, from the FDA. And then a big component of what we have been tasked with doing for all of these OWS run trials is to design the immune correlate study. And so I'll say a few words more specifically at the end about what immune correlates are and sort of the role we're playing and the interesting statistics that arise from those, the study designs that we're using to study immune correlates. And so that's the role that we all play, and anyone who's been involved in this effort, I think you'll see with sort of dark circles under their eyes, because it's been a kind of crazy few months of kind of endless phone calls and getting these trials off the ground as fast as possible while being as rigorous as possible. And so we've been sort of working through in that time a number of really challenging issues. And so this talk really, I'm just going to walk through a few of those issues. And again, I'm happy to hear input and feedback on this. And these are sort of really tricky issues where I don't think there's necessarily a right answer. And so I'm just trying to highlight some of the discussions that we've been having and sort of where we've landed on those and how these trials are being conducted. And so to start, I want to just kind of from a high level describe what these trials look like. And so I'll highlight the AstraZeneca design. So that's a company that I've been working with for the longest, but in broad strokes, all of the trials have a very similar design. So I'll walk through sort of what that looks like. And then I'll note that it's sort of unusual that there's been kind of a public pressure campaign that was underway a couple, I guess, gosh, time is a flat circle. I don't remember, maybe 6 weeks ago, 8 weeks ago, maybe 2 years ago, who knows. A while ago, there was a public pressure campaign to push these companies to make their trial protocols publicly available. And when I first started seeing this, again, this was like my first introduction to Twitter. I was like, oh, Twitter's cool. I'm going to tweet at this guy, Eric Topol. I don't know who he is. And so it's been kind of a wild ride. So Eric Topol is one of the big proponents of this, making these protocols public. And at the time, coming from academia, it was kind of like, we really don't make protocols public. I had no idea that was sort of standard practice in the field. And so it was kind of interesting that really in pharmaceutical world, these protocols are viewed sort of as a trade secret, as something you wouldn't want to make public. But anyway, the somewhat unusually, the companies have made these protocols available. And if you go to my slides, I have linked to the protocols that are here, and I'll update this, as Novavax and Sanofi's protocols become available as well. So you can read the protocols, and sort of it was an interesting case of everybody wanting these protocols, and then they got released, and everyone was like, "Wow, this is 100 pages of like really dry statistical and clinical language, and how do we make any sense of this?" And for people who aren't sort of familiar with reading clinical trial protocols, I think they were sort of having trouble making sort of heads or tails of that. So I'll talk a little bit about what these designs look like, some of the key issues that come up in the development of these protocols. Okay, so the big picture, all of the phase 3 trials are similar to this sort of design at the bottom of the slide. Again, this is AstraZeneca's specific design. And so the companies are recruiting adults. So I think Pfizer may have expanded their enrollment to include children over the age of 12, but for the most part, these are studies in adult populations, relatively healthy, stable, not in pregnant women. And really, we're trying to target individuals who are at increased risk for SARS-CoV-2 acquisition and COVID-19. And I think, the reasons for that are twofold, of course, right? There's the argument that these are the individuals who are, most adversely impacted by the pandemic, of course. So we would like to get a product approved in this population who's borne the brunt of the pandemic. And then, from a kind of cold-hearted statistical point of view, right? High-risk individuals are ones that accrue study endpoints the quickest. They're the ones that we can get an answer the quickest in terms of efficacy. So a lot of effort has gone into sort of screening individuals to enroll in these trials to try to recruit individuals who are at increased risk for acquisition. Okay, so after screening of individuals are randomized. So AstraZeneca in this case is randomizing 2-to-1. That's about split down the middle in terms of who's doing 2-to-1 versus 1-to-1 randomization. And then after randomization, there's some scheduled sort of follow-ups, and that's what's sort of shown here in this timeline. So the red lines are indicating a vaccine dose is given. So AstraZeneca is a 2-dose vaccine. I think all of them are except for Janssen. And you can ask Alex if I'm right about that or not. So all are about 2-dose vaccines with doses separated by about a month. And so in between doses, there's a scheduled phone call, right, where we're looking for adverse side effects associated with vaccination. There's an immune response measurement, which we'll talk about more when I get to the immune correlates part of the talk. And then after the second dose, so basically 2 weeks after the second dose is sort of when the clock starts for counting endpoints. And so we'll talk about what do I mean by endpoint pretty soon, but there's basically 2 mechanisms whereby we ascertain infection and disease. So there's sort of a passive follow-up that happens, right? So if you start coming down with COVID symptoms, you wake up and you can't taste your bowl of cereal, you give your study site a call, they tell you to come in for a sick visit, right? You get a PCR test and we check whether you are PCR positive for SARS-CoV-2. And if you have a set of qualifying symptoms and you come back PCR positive, you get adjudicated as a COVID-19 disease endpoint. So there's this sort of passive follow-up that's happening in these trials. And then there's also an element of active follow-up. So there's scheduled clinic visits, like at day 90 and day 180 in this case for AstraZeneca, where all individuals in the trial come in and they're given a serology test. And so serology is testing for antibodies. So it's basically checking whether you were infected in the past. And it's checking for antibodies that aren't associated with the vaccine. So we're able to distinguish here between the antibodies generated potentially in response to the vaccine and ones generated in response to natural infection. So at these times of active follow-up, we're actually able to ascertain asymptomatic cases, right? Individuals who never had any symptoms, so they never came in for a sick visit, right? But we see them at these scheduled time points and we're able to tell that they were at some point infected with SARS-CoV-2 between these scheduled follow-up windows. Okay, so that's the general sort of story of how these trials are designed. So 2 doses, 2 weeks after the receipt of the last dose, the clock starts for endpoint ascertainment, active and passive follow-up for endpoints. Okay, so what's the primary hypothesis test? So how do we measure how well vaccines work? So we do it with this sort of weird parameter called vaccine efficacy, or VE. And it's like 1 minus a risk ratio, which is sort of weird. We've learned that even the head of the FDA has trouble distinguishing between multiplicative and additive effects. So let's take care to carefully define what we mean when we talk about VE. So it's a percent reduction in relative risk when comparing vaccine to placebo, right? So it's this quantity, 1 minus a risk ratio, where I have risk in quotes here, right? Because I'm being intentionally vague right now in terms of how we're quantifying risk. But VE is 1 minus that risk ratio. So just to give you sort of a flavor of how do you get good VE, what is good VE? Well, a perfect vaccine, right, would bring your risk down to 0, right? And in that case, VE would be 1 or 100%. It'd be a 100% effective vaccine. On the other hand, what's a no good vaccine? Well, if VE is 0, what does that mean? Well, that means that the risk in the vaccine is pretty much equal to the risk in the placebo, which means the vaccine is not doing anything. So you get a VE of 0, right? That means the vaccine is not doing much. If you get a VE less than 0, right, of course, that means the vaccine is harmful. That would be very bad, right? If you would have higher risk in the vaccine than in the placebo, VE would be less than 0, okay? So why have I put risk in quotes here? Well, I have a couple questions still to answer, right? Risk of what? What endpoint are we talking about? Is it infection? Is it disease? Right? What do we mean by risk of what? We'll talk about that in the next few slides. And then also risk is in quotes because how are we going to quantify risk, right? And there's lots of different measures of risk, maybe one based on a hazard function, one based on cumulative incidence, like a survival probability, or one based on incidence rate. So there's lots of sort of different ways that, you know, we statisticians like to argue about in terms of how we quantify risk. It turns out because we're in sort of a rare event setting that all of these estimates end up looking somewhat similar. So it's really sort of not a point that's worth hampering on too much here. Okay, so what has FDA said about sort of how do you show a vaccine is good? What are they interested in? What kind of products are they interested in approving? So they have a guidance document that's linked here that you can go and check out if that's your bag. But I'll summarize the key points for you. So they said that in order for a vaccine to be declared successful, to have a shot at licensure, you need to qualify basically in 2 ways. You need to have a point estimate of VE for your primary endpoint. And again, we'll talk about what these endpoints are. So you need to have a point estimate for VE of at least 50%. Right, so basically cutting risk in half relative to a placebo. And you need to have the lower bound of an appropriately adjusted confidence interval be greater than 30%, right? So we have to rule out vaccines that are less than 30% effective, right? And our point estimate for VE has to be at least 50%. And we have to do all of this while controlling type 1 error at 2.5%. So that's what FDA has said they're looking for in terms of approving these vaccines. So how are these companies going about estimating testing VE? So there's, it's kind of a hodgepodge, right? So I sort of just took the protocols here and summarized the approaches. So, you know, I'd say Moderna, AstraZeneca, and Janssen are doing fairly standard things. So Moderna is just a Cox model. So they're quantifying risk using sort of a hazard ratio and reporting VE is like 1 minus a hazard ratio. AstraZeneca is using a Poisson regression, which is, you know, like a Cox model in disguise. Janssen has sort of an interesting sequential probability ratio test. And so they all sort of have their own analytic approaches. Pfizer's a bit of the oddball here. They're actually using a Bayesian approach with a sort of beta binomial approach where they're checking for posterior probabilities of VE being greater than 30%, given the data that they've seen. So they were sort of the oddballs, I think, though at the end of the day, sort of if you compare these different studies and their operating characteristics, from what I've heard across, you know, OWS statisticians is that there's really very sort of minimal difference between the operating characteristics of trials based on these estimation approaches. And this is something that, you know, sort of stressed us out as statisticians at first, is like all these trials using all these different approaches and sort of, I think we kind of arrived at the conclusion that really when it comes down to it, the data are what they are, and all of these approaches are essentially making the same inference in terms of a binary hypothesis testing decision, pretty much having similar operating characteristics, so not anything worth losing too much sleep over. The more interesting discussion, I think, is about, you know, sort of when we talked about quantifying risk, there's the statistical definition of risk, and then there's also what is your, you know, clinical definition, risk of what? What's the most relevant clinical endpoint to be studying for these trials? And so a large collaboration of statisticians who were working on these trials recently published a paper in Annals of Internal Medicine, the links here, where we were really discussing what are the potential options for endpoints for COVID vaccine trials, and what are some of the benefits and drawbacks of each. And so let's sort of walk through all of the different endpoints we could potentially adjudicate in this setting. So we know individuals become infected with SARS-CoV-2. right? So everybody who's infected comes here, and then those infections split, right? You either do or you don't develop symptoms associated with that. And we know that there's a fair proportion of individuals who never end up developing symptoms when they're infected with SARS-CoV-2. So we could classify them as having an asymptomatic infection, right? But of course, then there's some who qualify as a symptomatic infection, right? They have some lists that are checked off. CDC produced basically a list of symptoms that qualify you for having COVID-19 disease, right? So if you check these boxes, you get qualified as having COVID-19 disease. But even beyond that, right, we know that even people who are symptomatic, very often have very mild symptoms, right? So we could further categorize individuals having COVID-19 disease into being non-severe COVID-19, right? So maybe you lose taste for a week, you have a mild cough, right, but never really require any intensive intervention. Never developed extreme difficulties breathing or anything like that. So you would be qualified as non-severe COVID-19, whereas we know that some individuals are highly impacted by this, right? Go on to develop severe COVID-19 disease. And so which one of these, right, should serve as an endpoint for these vaccine trials, right? Which one is the most relevant? And so these are like BOD arrows. I think I'll come back to that in a couple slides to describe sort of one proposal that we put out there in this paper, which is to quantify sort of a burden of disease endpoint. So I'll describe that in more detail. But let's start with sort of the more clinical measures, right? So SARS-CoV-2 infection. Okay, so why might we like just any infection, right? Why might we like that as an endpoint? Well, of course, it's relevant in the context of a pandemic, of course, right? If you prevent infection, you prevent spread of the virus, and you help stem the pandemic. Right, so that's one reason that we might like that from a public health perspective, of course. But one reason from a statistical perspective, right, is that also that we'll observe many more infections than we will cases of clinical disease, right? We know that there's a high proportion of individuals who, when infected, never go on to demonstrate symptoms. So just thinking about a statistical power angle, right, will be well powered potentially to reject a null hypothesis about a VE against SARS-CoV-2 infection. Of course, the downside here is what's the clinical significance, right? Is you're sort of mixing up very severe clinical cases, right, with ones that never go on to develop any symptoms whatsoever. So there's a question of clinical relevance of this endpoint. Moreover, there's sort of some statistical challenges associated with this as well. So of course, we're only capturing asymptomatic infections at these passive follow-up visits. So we sort of have this course measurement in time. There's a potential for many false positives, right? Remember, we're going to be testing all individuals in the trial, and it's this sort of classic Bayes theorem example that you get in every Biostat 101 class, that even if you have a test that has high sensitivity, high specificity, but low prevalence in the population you're doing the test in, right, there's a potential for a high false positive, potential for low positive predictive value, right? And so what you can show is the impact of that on your estimates of VE will be biasing VE towards the null. And so it's sort of a statistical challenge when your endpoint is being measured with error, right, to sort of correct for that and draw a valid inference. Okay, so there's sort of upsides and downsides to that infection endpoint. Similarly for COVID-19 disease, right? So we're certainly approaching something that's more clinically relevant, right? Because we're ruling out all those infections that never result in symptoms. Moreover, we expect there to be a reasonable number of cases, at least relative to severe COVID, right? More individuals will develop mild symptoms than severe. And so we'll be still, hopefully we can design trials that are reasonably powered to detect VE against COVID-19 disease. But then there's still a question about clinical relevance, right? So we're still mixing up individuals who experience mild symptoms with those who are, requiring, ICU stays, requiring ventilation, right? So are we really targeting the most clinically relevant endpoint? So the most clinically relevant endpoint then, of course, would be severe COVID. Right, and moreover, this is sort of the endpoint for which we would maybe a priori expect the highest efficacy, right? So there's sort of a well-trod tale in vaccine development of vaccines that don't necessarily prevent infection, but they do lessen the severity of disease. So you end up with quite a high VE against severe disease and potentially a lower VE against infection or more mild disease, right? So high a priori expected efficacy. The challenge of this endpoint, of course, is that there's very few cases to be expected. And so we would need to do either larger clinical trials or have follow-up last for a longer time in order to be well powered to detect VE against severe COVID. And so maybe I'll just say a few words about this burden of disease endpoint. So we propose this sort of burden of disease endpoint, which if you see here we sort of, basically we score each endpoint, right? So we score it according to the clinical burden associated with that endpoint. So if you're an asymptomatic infection, you get a score of 0. If you're symptomatic, you get a score of 1. And if you have severe symptoms, you get a score of 2. And then you can define sort of VE as a proportional reduction in the score associated with infection. Right, and so this is certainly more clinically relevant than COVID because you're kind of upweighting the more severe cases. And that's good. And it has some nice operating characteristics that we describe in that paper. But sort of course, the controversy of assigning a score is that whatever score you pick is going to be arbitrary. Right? So what's the best way to assign a burden score, right? Why should severe disease only be twice as bad as non-severe disease? Shouldn't it be 3 times or 4 times or 5 times? What's the appropriate scale? And so discussion of this endpoint, unfortunately, tends to get bogged down into these specifics, or you get a review back from the statistical reviewer at New England Journal that sinks your submission because they said you're, quote, treating an ordinal outcome as continuous, and that's a big no-no. And so that's sort of where we're at with this disease. Is I think a lot of the statisticians are sort of on board with this being sort of an interesting endpoint, but it's challenging to get buy-in from others in this context. Okay, so right now, FDA guidance is saying that either COVID-19 disease or SARS-CoV-2 infection is an acceptable endpoint. So that's what their guidance document says. Then for whatever reason, as far as I can tell, OWS has been telling companies that you can't look at just infection alone. You have to consider clinical disease. So the result of that is that most companies have settled on COVID-19 as the primary endpoint. So any symptoms that qualify according to CDC guidelines in PCR confirmed SARS-CoV-2 infection. So that's the primary endpoint in almost all of the OWS trials, the exception being Janssen, which is going for a severe COVID endpoint. And as a result is having to recruit twice as many participants to be well powered against that endpoint. So I'll say a little bit about monitoring since time is running low, and I promised I would show you math, so I've got to get to my math. So sequential monitoring has been something that has been very sort of controversial and since the release of these protocols, and some big questions have been raised about whether sort of the monitoring that's being employed in these trials is too aggressive, or whether we're really going to accumulate enough data before one of these companies potentially submits for an EUA. And so I kind of point out here the interim monitoring plans that are associated with each of these protocols. Sort of, they're waiting for a number of endpoints to accrue. So Pfizer will do their first interim analysis after the first 32 endpoints. And then again after 62 and so forth. And so of course, the more times you look at the data, we know that the more times the greater chance you have of falsely rejecting a null hypothesis. So you need to do some kind of alpha adjustment. So all of these sort of fall into one of the two classic camps. Pfizer's stopping boundaries end up looking very much like POCOC. Moderna and AstraZeneca are explicitly using O'Brien-Fleming. And Janssen's, while not called POCOC, end up looking something like POCOC as well. And so there was, again, a lot of public criticism about this, sort of saying, if Pfizer's trial is only run long enough to accumulate 32 endpoints, right? Is that enough? And sort of on the one hand, from my perspective, I can look at the math and I can say, yeah, we're controlling type 1 error and that's good. And on the other hand, I can my feeling as a human is kind of like, wow, we're going to vaccinate, 5-6 billion people on Earth based on data on 32 individuals. And so for me, there's a real dissonance kind of there where I sort of agree with the public perception that makes me feel very uncomfortable that could potentially be the case, even though, statistically it's a valid thing to do. And so in response to this criticism about these interim monitoring plans, FDA has sort of beefed up the requirements that they've said that they're going to need for an EUA. And this is sort of a moving target. So I actually, this is changing day to day. And I hear mostly about updates to this from reporters more than I do from my colleagues in OWS and at these companies. So it's a moving target. But what FDA has recently said is that they may require, in order to grant an emergency use authorization, basically some further stipulations. So at least 50% of participants completing 2 months of follow-up, right, to make sure that we've accrued enough safety data. And then at least 6 COVID cases in the oldest age category to make sure that we can have some chance of detecting efficacy in older adults. And at least 5 cases of severe COVID in the placebo arm, so that we can at least hope to have some signal to talk about VE against severe disease. So I'll skip this slide. I'll just say that sort of, I did some work to show that Pfizer stopping boundaries look like a POCOC, and that's on GitHub if you want to check that out. I'll skip these slides. So just briefly, if you want to end up going through these slides, I kind of give my opinion on what's, what happens if efficacy is declared early? And that's the big question, right? What happens if Pfizer looks at their data 2 weeks from now and they've hit their success criteria, right? Should we feel good or should we feel bad about that? And I think there's sort of reasons for both, right? If we should feel good because the vaccine will probably be available to you sooner, or at least to your most vulnerable friends and relatives. And moreover, right, just statistically based on the data, right, if Pfizer declares success after their first analysis, well, then there's a really good chance that their vaccine is working pretty well. There's, of course, uncertainty in that, right? But you can sort of convince yourself that if you're looking at the first 32 cases and you end up with a 26-6 split placebo versus vaccine, that's a pretty good vaccine. And so that would be good news. On the other hand, the big question is what happens to the other trials? And this is what we're really grappling with right now in OWS is what will the other trials look like if one of these companies is granted an EUA? What will we have to do? Will we have to discontinue follow-up? Will we have to offer placebo recipients that vaccine? How's that going to work? And so that's sort of a big ticket item that we're working through right now. And so I'll just very quickly, since we're running short on time, say a few words about vaccine correlates, because this is a big area that OWS and the group at the Hutch have been working on developing an analysis plan and a sampling frame for. So what are correlates of protection? Well, Peter and Stanley Pott can put down some nice language to describe what those are here. But basically, we can think about a correlate protection as potentially being a candidate for a good surrogate endpoint for vaccine efficacy, right? So what we're looking to do is find an immune response in an assay to measure that immune response, right? That's reliably predictive of how well vaccines work. And why is that so important? Right? Well, it can accelerate the approval process, right? It opens up a regulatory pathway potentially for approving vaccines based only on their impact on an immune response, right? So it's important for speeding up the development of vaccines, for the approval of vaccines, as well as potentially accelerating approval for licensed vaccines moving to different populations, right? So we talked about how most of these trials are only enrolling adults. Well, what if we need to use the results of a phase 3 trial in adults to bridge to children? Well, good correlates of protection could help us with that bridging process without needing to go through a large phase 3 trial in children. So a potentially speedy pathway for vaccine development and approval of new vaccines as well as existing vaccines in new study populations. And so what is the sampling frame that we're using to evaluate these immune responses? And so if you'll remember way back when I showed you a design of the AstraZeneca trial where we're measuring immune responses, we're doing a blood draw at least on all the trial individuals after the receipt of both doses of the vaccine and actually longitudinally over time after that. And so you could say, okay, great, we can go ahead and assay all of those blood draws, right? And so these trials are about 30,000 individuals, up to 60,000 individuals. And so we could do that, right? But it would be very expensive, potentially timely. And as it turns out, it's sort of statistically unnecessary to do that. We're actually able to get good power for detecting immune correlates, assaying far fewer than the full 30,000 participants. So how do we do this? We use case cohort sampling. And so how this works is we basically take a blood draw on all individuals of the trial, but we store that blood, right? And so a certain number of individuals in the trial population are sampled into this correlate subcohort. So this is the cohort part of the case cohort design. So we're grabbing a random subset of individuals and about 1,600 of them in this case. So that's our cohort. And we measure immune responses in those individuals. And then we go back and we augment the data that we have by measuring immune responses in everyone who ended up being an endpoint. So anyone who ended up infected or a disease endpoint, we'll go draw their blood, or we'll go retrieve their blood samples rather and assay the endpoints, the immune responses as well. And so remember, when we're thinking about binary trials or trials with binary endpoints or survival endpoints, right? It's really the individuals who experience the study endpoint that are driving power. Okay, and so what we're doing with this design is ensuring that we're capturing all of those endpoints in our data and augmenting them with this random cohort, right? So that's how we end up getting approximately the same power as we would have if we had assayed the blood of all 30,000 individuals in the trial. Okay, we made it to the math with not 5 minutes to spare. So let's chat a little bit about math. Am I getting a Okay, just a couple comments in chat. Okay, so we'll take questions in chat after this is over. So okay, gotta get to the math. Okay, so here's some quick notation. So this is just kind of highlighting one of the challenges in doing statistical estimation when you have case cohort sampling. And why is it challenging? Well, remember that we're not taking a fully random draw of the population, right? There's some bias in the way that we're sampling these individuals. In particular, when we augment the data with these COVID cases, well, the individuals who end up being infected or having clinical disease are likely different than a random individual from the population, right? So we have a biased sample. So we need to think of some way to correct for that bias, right? And so we can do that using sort of classical technique. So here I'll just outline a little bit of notation very quickly and show you some very boring estimators. So if we think of W as being these baseline covariates, so these are things like your age, your race, ethnicity, and so forth. A is your vaccine assignment, so that's a binary 0-1 deal. Y is whatever this COVID endpoint we're interested in studying is here. Maybe it's infection, maybe it's disease. And then we're going to introduce a couple of new variables, right? So we have this delta. This is the indicator of being in this immune response subcohort, right? So did you end up having your immune responses measured, either as a member of this random subcohort or as a case? Right, and amongst those individuals who do have delta measured, we get to see S, their immune response. Okay, and now we have some parameter of this distribution of these random variables that we want to estimate. So we can take a very simple parameter, very uninteresting parameter for the purposes of assessing correlates, but one that's nice for illustrating these ideas of how we correct for bias. So suppose we're interested in just estimating the overall average immune response, right, the mean of S. Okay, but our data are subject to this case cohort sampling. So you could say, well, I've taken an intro stat class. I've learned about the Horvitz-Thompson or inverse probability of treatment weighted estimator. Maybe I could do some inverse weighting here to do that. And so indeed you could, right? So we'll just basically take the values of S, right? And we'll multiply them by this inverse probability weight where we're weighting by the inverse probability of being sampled into the subcohort, right? And that's of course something that we know by design. Because we control that probability. If you had an endpoint, this probability is 1. If you didn't, it's whatever your probability is of ending up in the random subcohort given your covariance. So we have this simple inverse weighted estimator, and we can develop a simple sort of Horvitz-Thompson IPDW estimator based on that. It turns out that there's more efficient ways to go about this, of course, by augmenting the IPDW estimator. And that estimator is shown here. So it involves the IPDW estimator plus a little extra, right? And this little extra piece in particular involves some regression. So this regression quantity highlighted here, right, is a regression amongst individuals who you measure the immune response in. We're going to fit a regression where the outcome of that regression is the immune response regressed against their vaccine status, covariates, and their endpoint status. Okay, and the neat thing about this, right, is that this estimator turns out to be doubly robust. So even though we have to do a little bit more work, we have to fit this regression model. Some people might say, well, what if I get that regression model wrong? Well, one of the nice things about these augmented approaches is it turns out it doesn't really matter in terms of the consistency of the estimator. You can still recover a consistent estimate of your parameter of interest, even if this quantity is inconsistently estimated. So it's sort of low-hanging fruit in terms of improving efficiency at little risk of validity. And so this is a very boring parameter. Of course, we study more interesting and complex parameters when we're actually doing these immune correlates. But this is the sort of game that we have to play when we're thinking about crafting these statistical estimators. We need to do, take an existing estimator, maybe a simple one, right? And think about how do we need to update that estimator to account for this case cohort sampling. And so we have a nice paper that a student that I work with at Berkeley just had come out in biometrics that shows how you can do this for kind of a causally flavored parameter. That's really ugly and it's shown here, but let's just describe what it says in words very quickly. We're basically interested in understanding the counterfactual risk of COVID if we give you a vaccine and we also increase the immune response associated with that vaccine, or we decrease the immune response associated with that vaccine. So we have this little delta guy, and that's saying we're going to take the vaccine immune response that the vaccine induces, and we're going to either shift it up or shift it down. And we're going to study the impact, the causal impact that has on your risk. And so this is the sort of figure you end up with for that. So at 0, when delta is 0, we're not shifting your immune response at all. So this is just sort of the observed risk of individuals for the disease endpoint in the vaccine arm. And then we're saying, what happens if we remove a little bit of that immune response? That's what's happening when we're moving to the left here. We're taking away a little bit of immune response. right? And if that immune response is important, right, we'd expect that that risk would increase, right? And so that's sort of what we see. This is an example from an HIV trial. And so we have a nice way of sort of estimating this efficiently using these augmented strategies and potentially summarizing this curve over delta using a working model. And so I'll refer you to this paper. It's linked here if you're interested in hearing more. So I'll take just maybe 1 minute to say current status, everything's in overdrive. I haven't slept in months, and we're working to get a correlates analysis plan out as soon as possible. Let me conclude though with a few sort of thoughts for the general public, some takeaways from the state of COVID vaccine science. And, you know, in the current political climate, right, there's probably rightfully a huge amount of distrust of the federal government. And unfortunately, the regulatory pathways for vaccines go through the federal government. And so naturally, there's this distrust sort of shifted onto this entire COVID vaccine development enterprise. So I would just conclude by saying from where I sit, the science behind the development and evaluation of these vaccines is extremely rigorous. And if I could have thought of a group of scientists that I would want overseeing these trials in terms of who's working with OWS, who's sitting on these DSMBs, right? They're the right people in the right places right now. So from where I sit right now, science looks good. And also these are individuals that if the science starts to look bad, if political interference starts to happen, right, you're gonna hear from them, right? These aren't all federal employees. Our hands aren't tied to raise red flags in terms of things going in an unfortunate direction. So I would say that, you know, increasing transparency in the form of releasing these protocols and potentially other avenues that we can think of is good, right? I mean, we're all academics, we like open science, so that increased transparency I think is great. And then the final point I would make is that, you know, sort of I've alluded to, there's really hard questions that we've been faced with in designing these trials. And oftentimes there's no right answers, and sort of there's room for healthy academic debate, I think, in this. And I would say that one of the challenges right now is that a lot of this academic debate is taking place in atypical venues. It's happening on Twitter, it's happening in op-ed pieces. And I worry a little bit that the result of that is increased distrust in this process. And so I think we as academics should be brainstorming ways when we're in a situation that's this fast moving, right, and these debates need to happen, how can they happen? How should we think about the language that we use to sort of have these debates in a way that doesn't increase distrust, right? Because for a general member of the public, they see people saying completely opposite things. The conclusion might be, well, nobody knows what they're talking about, and this is all BS. So I think we just have to be a little bit careful in having these debates and the language that we use and addressing that a lot of these questions are very gray and nothing is really black and white. So I'm really grateful for this opportunity to sort of give you this high level overview. I'm really grateful for the great collaborations, you know, my time at UW, establishing these collaborations because they put me in a place to do really cool science going forward. So really glad to be giving this talk and happy to stick around and catch up with anybody who's here. And please do reach out if you'd like to follow up about anything. Thanks.
Thank you, David. Great talk. Yeah, time for questions. I think you can read them, so maybe you can go through them yourself.
Okay, let's see. Scott.
Scott has raised his hand, so if he wants, I can unmute him. And
oh, we have the power.
Maybe we'll wait for that.
Sure, Scott. I see you have a question in chat, but if you want to, you know, ask another one, please do. So do I unmute him? Scott, you're allowed to
lowered his hand. So
there we go. Now?
Yes.
Yeah.
So it relates to way back when I asked this question was about the endpoints and the sequential stopping rule, as well as your correlates. And that is the focus on calendar time. The time, you spoke to it a little bit of durability, but to me, the advantage of being just a little bit slower ensures that. And so the snarky question I'll ask is, if we took the response with the dengue virus based on the first 32 events, what would be the finding of the correlates versus what we now are concerned about with the dengue virus? So in other words, it's this idea that a waning effect can cause great problems.
Yeah. Yep. Yeah, you know, I'm a statistician, Scott, so right, more data is better, right? But I would say, you know, the calculus changes a bit in the context of the pandemic. I'm not saying it totally shifts away from, you know, granting an EUA after 32 events, because of course we care about durability. We care about waning immunity. We care about potentially enhanced immunity in the context of waning antibodies, or sorry, enhanced risk in the context of waning antibodies. So yeah, there's really important questions, but at the same time, sort of, there is a pressing need to, you know, help control the pandemic. So
But
it's
your
an
slide,
impossible value proposition is where I'm at. So I
your
don't know
slide,
who's qualified to answer that.
your slide about acceleration was a very nice one, to say really what we're doing is we're saying it's worth wasting money on things that might not pan out, so that whichever one pans out we can go with. But and personally would accept taking the vaccine every 3 months, but not much less than that. You know, so I would hate to accept a vaccine where the efficacy was less than 3 months and there was another one out there that was worthwhile. And again, the unproven, so and I'm hitting up mostly, most of Eric Topol's comments I think were well-based, although he didn't put he put too much statement of the sequential monitoring was bad rather than what the time frame. If you read the rest of his arguments, they were okay. But the particularly with a DNA or an mRNA vaccine,
Right.
To be going out on the limb with that is extremely problematic.
Yeah.
When we don't even know that with so few events, and if all we're talking about is another couple months of follow-up, that really doesn't slow us down much.
Yeah.
So that's my major.
I completely agree. I mean, it's challenging. I think FDA is starting to move in the right direction now in terms of thinking about what else they're going to require and whether there's other mechanisms besides an EUA that they're going to encourage companies to go for.
But and just the last comment is, none of us believe in the proportional hazards model. And as you did correctly state, you know, whether you do negative binomial, whether you do Poisson regression, whether you do, by the time you use any sort of a robust standard of error, they're all doing the exact same thing. You can't find a dime's worth of difference between them. So that means they're all subject to this problem with a time varying effect that we just need to be very nervous about.
Yeah. I mean, it's at the end of the day, I think, you know, you can summarize VE at a given time point, right? But that's not going to tell the whole story in terms of what's clinically relevant. Yeah. Thanks, Scott. Nice to hear from you.
Well, it was a very nice talk. Thank you. I haven't interested in what's being done here.
Thanks. See a question from Navneet. Let's see. Given all the current COVID trials and plans for additional trials, there's likely competition for recruiting trial participants. Is there a way to quantify this competition? And two, would it be straightforward to add this information to your augmented estimator? Yeah, it's a good question. I think we are running the risk, I think, a little bit of depleting participant pool. You know, I'm not close enough to the recruitment and the ongoing studies. I'm not involved in Moderna or Pfizer, obviously. So to say what that effort has sort of looked like. And to answer your question, would it be straightforward to add this information in your augmented estimator? So I'm going to do the political thing where I take your question and I answer a slightly different question that I think is interesting. So what I would say to that is, one of the concerns that I have is sort of this game theory that exists, that's unspoken, right? That FDA has encouraged these trials to recruit, you know, X% over a certain age, X% from a minority group, right? And particularly with the age one, right? Because it's challenging oftentimes to get vaccines that work well in older adults, because the immune responses just work a little bit differently there. And so there is sort of this game theory that I worry about that says, like, what do these companies have to do to convince FDA that they tried hard, right? That we tried hard to recruit older adults and we just didn't get that many, right? But then maybe some other company, right? Like, so maybe Pfizer tried their hardest and they only got 20% over 65. I think their enrollment stats are out, so maybe this is unfounded, right? But then one of these OWS trials recruits a much higher proportion of individuals over 65, and therefore is at a disadvantage when you think about overall efficacy, if it turns out that efficacy is lower in older adults. And so when it comes time to compare these vaccines head to head, I think it'll be quite interesting to think about ways we could standardize, you know, these vaccine effects to get a more fair comparison. And so I do think there are nice ways that you could do that using augmented estimation approaches. So again, not quite your question, but one that I think is pretty interesting. And you're muted, so not mute, so you can't complain, right? So Ernesto, I see. Clinically, asymptomatic cases may not be too relevant, but they are in terms of controlling the pandemic due to asymptomatic transmission. Does this come up? Yeah, it definitely does, right? Because obviously asymptomatics are highly important. And so there's this sort of, again, impossible proposition of weighing the benefit to public health and stemming the spread of the pandemic versus, choosing a clinically relevant endpoint. And so that's sort of where I think what we've recommended in that Annals paper is that we need to be reporting on all these events, obviously. But in terms of what goes through as a primary endpoint for licensure, COVID-19 disease seems like a reasonable sort of middle ground endpoint, but we do like this burden one as well. And sort of, it's interesting, I don't know anything about modeling these things in terms of what vaccine impact will be, but I've been speaking with some people who do think about that quite a bit. And they have a very different perspective in terms about how they would project out, the impact of this vaccine. And it's more of a public health perspective, right? Where they're thinking about what is the effect on asymptomatic infections? What impact does that have on transmissibility? What is VE against transmissibility, right? Can we measure that, right? In terms of viral shedding or another endpoint. So yeah, so there's a lot of questions, that aren't even captured just by that brief endpoint discussion that I have there. All right, a question from Marlena. Has your experience working on Warp Speed Initiative led to new insights about how to speed up trials for other applications that aren't COVID? Honestly, I haven't paused to think about that too much. It's a good question. Yeah, I think probably we'll have to take stock of that when I stop being on 12 phone calls a week and needing to get SAPs published as soon as possible. But yeah, I think there's going to be sort of a, not just for vaccines, but treatments and, and other prevention across all public health, right? Like we saw a little bit with Ebola in terms of a reckoning of the field and thinking about it's just when we have a moment to pick up our heads, catch our breath to say, what did we do? What could we have done better? What can we learn from this? So I personally am not at a moment where I've had time to reflect on that, but I think the field will be there hopefully pretty soon.
Yeah, there's another question from Scott, I believe.
Just in response to that, we recognize that we are lowering our standards by your thing, that we are accepting a level 0.025 from a single pivotal trial.
Right.
Rather than requiring a couple. So we need to, that we are doing a lot to recognize that we'll put up with some more false positives in this pandemic situation, hoping that we will be able to correct them. And by the fact that we have multiple vaccines going, the worst thing that would happen to my mind is if some vaccine gets approved and we ruin the other trials that are going on at the same time.
Yeah, no, and I have that point in my slide. So a great point about the single pivotal trial. Of course, yeah, that is that we are lowering that phase 3 burden. But yeah, I think the worst possible outcome here is a middling vaccine sneaks through, and then all the other trials are impacted, and so that's sort of what we're grappling with. And I think, we're coming up with arguments as to why we should be able to continue placebo-controlled trials and blinded follow-up, but it's going to come down to seeing whether participants are willing. Right, and as well as availability, and what that looks like will have an impact. So we're sort of preparing for 100 different scenarios and have no idea which one we'll actually end up in right now.
Thanks, David. So I heard that the human challenge trial was approved in the UK. What's your thought on that, and how much that can speed up things?
Yeah, that's a question a lot of people have been asking. I'm not sure. I think on the one hand, we like challenge trials. Of course, they can help, validate a correlate. They could potentially, be a mechanism for approval. I'm not sure what regulatory agencies say about that. I'm not sure that it's discussed in FDA guidance. What concerns me about it, of course, is that by design, these trials have to be conducted in, the healthiest populations. And so I have some concerns, I guess. You better believe that correlate is good such that you're able to bridge the results of those studies happening in young, healthy adults to, you know, the individuals that we really need to get this vaccine to, which are older adults. And so I'm woefully ignorant of sort of that process and what that could look like, but definitely something that the people in the field are thinking about.
Thanks. Yeah, I don't see any other questions. So yeah, I guess that's it. Thank you very much, David.
Of course, yeah.
We really enjoyed. There's also a couple of complimentary comments that I'm sure you've read. And
Yeah.
thank
Yeah.
you.